Hi there, I'm Shin Hatou, the CEO of our company Cellusion. Cellusion is a startup company from Keio University School of Medicine. My background is ophthalmologist, and my specialty is corneal transplants and the regenerative medicine of the cornea. Today, I would like to introduce our project, Corneal Endothelial Regenerative Medicine with iPS cell-derived endothelial cells. Here's the agenda. First, I'd like to explain our purpose of the project. And next, I would like to show you the characteristics of CLS1 endothelial cells. And next, moving on to the development schedule, competitive advantages, and business model. And finally, I would like to introduce our management team. There are unmet needs in corneal transplants. It is reported that more than 30 million patients are waiting for corneal transplants globally. However, only 180 corneal transplants per year are conducted. So the demand is 70 times the supply. And here you can see the clouded cornea. This is the bullous keratopathy, the corneal endothelial cell dysfunction. Bullous keratopathy is the most common disease for corneal transplants. More than 50% of corneal transplants are for this disease. Corneal endothelial cells are the innermost layer of the cornea. The causes of bullous keratopathy is corneal endothelial cell damage by surgical complications of intraocular surgeries or genetic abnormalities such as Fuchs corneal endothelial dystrophy. Current treatment for bullous keratopathies are penetrating keratoplasty or endothelial keratoplasty. Although the surgical procedure has been advanced, there still be many complications, and they are still very difficult surgeries. So what's our proposed solution? Our solution is corneal endothelial cell substitutes from iPS cells called CLS1 cells. We can produce corneal endothelial cells from allogenic iPS cells with mass production, standardized quality, and stable supply. Also, we have developed the cell transplant technique. Suspended CLS1 cells are injected into the cavity between cornea and lens called anterior chamber space. This surgery is very low in invasiveness, easy of handling, and ready to use care. So here you can see the preclinical efficacy study of CLS1 in monkey bullous keratopathy model. Top left, you can see the corneal edematous thickness from baseline. CLS1 transplanted eye in blue significantly reduced the corneal edematous thickness compared to the negative control eyes without cells. At the same time, CLS1 transplanted eye dramatically recovered its transparency compared to the negative control eyes without cells. Also, we have confirmed that the transplanted CLS1 covered the posterior surface of the cornea. So, CLS1 transplant reduced corneal edema, recovered corneal thickness, and recovered corneal transparency dramatically. So what about the production process and characteristics of CLS1? Left, you can see the production process of CLS1. Frozen iPS cells are thawed, cultured, and expanded. It takes 3 weeks for expansion culture of iPS cells. After that, CLS1 cells are differentiated from iPS cells with attached culture without passage. It takes just 2 weeks. And finally, CLS1 cells are preserved as frozen stocks. The cells are preserved until cells are used. Right, you can see the CLS1 immunostaining of corneal endothelial cell markers. The CLS1 cells showed monolayer morphology with cobblestone appearance. CLS1 cells express pan-function NAK ATPase, touch junction marker GO1, N-cadherin, and transcription factor PITX2. These morphologies are quite similar to the corneal endothelial cells. And also you can see the flow cytometry analysis at the bottom. Flow cytometry analysis of functional corneal endothelial cell markers, including N-cadherin as barrier function, NAK-ATPase and mineralocorticoid receptor as pan function. And you can see quite high positive expression rate of these markers in CLS1 cells. And how about the purity of CLS1? Top, you can see the immunostaining of undifferentiated cell marker OCT4, OCT4. iPS cells express OCT4. By contrast, CLS1 cells don't express OCT4. And bottom, you can see the undifferentiated cell population of CLS1 by OCT4 qPCR analysis. Undifferentiated cell population of CLS1 is below 0.01%, which is indicated by red dotted line. And also, you can see the cell growth analysis, which indicates there are no cells that proliferate abnormally in CLS1. So the purity of CLS1 is above 99.9%, and the cells, CLS1 cells don't include the cells that proliferate abnormally. So here you can see the brief summary of CLS1 preclinical safety studies. We have also conducted the in vivo tumorigenicity test, that is cell transplant into the animal anterior chamber of the eye. This method is also patented by us. And it is indicated that there were no tumorigenicity nor teratoma formation of CLS1. Also, we have conducted residual rate of undifferentiated cells, as I mentioned before, and genetic chromosomal abnormalities, general toxicology, and pharmacokinetics. And we have confirmed that there are no safety concerns in preclinical studies. So what's the CLS1 target disease? CLS1 targets mild and moderate bullous keratopathy patients as a first-line therapy because of the safety of the surgery. So let's move on to the CLS1 development plan. There are 3 points we would like to highlight. First, investigator-initiated clinical study at Keio University Hospital is scheduled at 2022 this year. This is a first-in-human clinical study. After that, Japanese phase 1-2 clinical trial is scheduled in 2023. This is a Cellusion-initiated clinical trial in Japan. Thirdly, the phase 1-2 clinical trial in US is scheduled in 2025. So now we are preparing for, now we are communicating with PMDA, Japanese PMDA, and also we are preparing for the FDA consulting and CDMO tech transfer globally. So what about our competitive advantages and IPs? We can produce CLS1 endothelial cells from allogenic iPS cells in just 1 month by our unique direct differentiation induction method. Secondly, we can treat more than 1,000 patients per batch. And thirdly, we can preserve the CLS1 cells as frozen stocks, which allows us to distribute the cells to the medical institutions globally. These competitive advantages are totally protected by 6 patents. Okay, well, what's about the competitive landscape? We are the pioneer of iPS cell-based cell therapy for bullous keratopathy. Although there are some competitors who use cultured corneal endothelial cells from donor cornea, however, the cell number from donor cornea is very limited, and the cell quality of the cells varies. Cell quality varies due to the donor variation. By contrast, we can mass produce the CLS1 cells with stable quality. And we also are able to preserve the cells as frozen stocks, as I mentioned before. So what's our business model? We Cellusion produce the CLS1 cells in CDMO and distributes the cells with pharmaceutical wholesale to the common medical institutions. General ophthalmologists can treat the global bullous keratopathy patients with CLS1 cells in common medical institutions. Also, its cost will be acceptable for insurer because of the mass production and the preservation. So how about the annual new patient forecast? The annual new patients number are forecast to steadily grow around 1.2 million per year, which is translated into $5.8 billion market annually. It is due to growing countries' medical level, increase of intraocular surgery and surgical complications, and population of aging globally. Okay, then next, let's move on to the launch schedule. We will launch the cells in Japan in 2026, in US in 2029, and the rest of the world in 2030. Okay, finally, I would like to introduce our management team. Again, I'm Shin Hatou, the CEO of our company, and Joji Hayashida as COO, formerly at Astellas Pharma, Michihiro Hayashi as CAO, formerly at MBL and experienced IPO, and Nobuyuki Imai as CDO, formerly at Kyowa Kirin Pharma. And this is the take-home messages. We are the pioneer of iPS cell-based regenerative medicine of cornea. We can mass produce CLS1 endothelial cells from iPS cells with stable quality and preserve the cells. We will treat bullous keratopathy patients globally with our cells. Okay, that's all. Thank you very much for your kind attention.